Ladies and gentlemen, the event will begin shortly. We kindly ask you to obey the rules during this event. Specifically, participants are required to dress neatly and politely. Participants are required to turn on their camera and turn off their microphone. Participants are recommended to set the speaker view throughout the event. Participants should not carry out any activities that are not related to this event. And participants should avoid any kind of distraction that could disrupt the course of this event. Your cooperation and patience is highly appreciated. The Honorable Dean and Vice Dean of Faculty of Pharmacy, Universitas Padjadjaran, Dr. Hien Duong, Professor Mary Jo Anety Corpus, PhD, Mr. Raymond, PhD, Dr. Sriwidodo, Dr. Ida Muspiro, Dr. Ian Sopian. All professors, faculty members, distinguished guests, and participants present today. Welcome to the second day of 4th International Seminar on Pharmaceutical Science and Technology 2020. Honorable guests, speakers, participants, ladies and gentlemen. The first keynote speech for today will be delivered by Dr. Hien Duong and will be led by Dr. Ida Muspiro. First, please allow me to read the curriculum vitae. Dr. Ida Muspiro, MSc Apotheker, has research interests in pharmaceutical analysis and medicinal chemistry. One of her publications is QSAR study of acetic acid and its derivatives as potential synthase inose inhibitors. With all due respect, we now give the time to Dr. Ida Muspiro.
Okay, Bismillahirrahmanirrahim. Thank you to Mr. Ceremony for the introduction. Good afternoon to Dr. Hinduang.
Hello.
And to all participants, good morning and Assalamualaikum warahmatullahi wabarakatuh. Hello. Hello, Dr. Hinduang.
Hello. Hello, everyone.
To all of you, welcome.
Yeah.
Hello. To all of you, welcome to second days of the. Yeah. This morning in the Bandung and maybe in Sydney already afternoon.
Yes, exactly. Already 12:30. Yeah, 12:33.
Okay. Okay, welcome to second days of the 4th International Seminar of Pharmaceutical Science and Technology. And I would like to welcome today's keynote speaker session, Dr. Hien Duong from University of Sydney. Once again, good afternoon. How are you today, Dr. Hien Duong?
Yep, good. Thank you. So I'm looking forward giving the talk. Yeah.
Okay, thank you for your time, Doctor.
Yeah.
Before we begin Dr. Hien Duong presentation, first I will present Dr. Duong curriculum vitae. Dr. Duong was awarded a highly competitive and for postgraduate scholarship and Joint Center of Advanced Macromolecular Design in 2007 for her PhD. In 2016, she joined the Institute of Biomedical Material and Devices, School of Mathematical and Physical Sciences, Faculty of Science, University of Technology, Sydney as a senior research fellow. And recently, Dr. Duong took up the position of lecturer in pharmaceutical sciences at the University of Sydney. And Dr. Duong has published more than 40 peer-reviewed journal articles relating to using smart nanomaterial for drug delivery. And Dr. Hindwang, your presentation time is 30 minutes, and then we will going to continue to discussion.
I'm going to share my screen now.
Yes, you can know, and I will inform to all participants. Please submit your question in the chat room, and don't forget to write your name and institution. And you can press the raise hand to ask directly for Dr. Hindwang. The time is yours.
Yeah. Can I confirm with you, like, you can see my slides?
Yes, I can see.
You can see my slide, and also you can hear me well?
Yes. Yes.
Yeah, perfect. Yeah, great. So first, I would like to thank you so much for giving me this opportunity to be here. That means be in front of my computer, not in Bandung.
Yes, because pandemic COVID-19.
Exactly. Yeah. So and I will give my presentation on my research. So as from the title in this slide, so you know that my research, maybe I would like to put it in the bigger screen. So give me a sec. I want to make it in the presentation mode.
Okay.
Yes.
Yeah, so already minimized.
Okay. Yes, it's a little bit small. So give me a sec. Maybe I stop sharing for 1 sec and so. Is it too small just now, or is okay for you? But
the presentation is closed.
It's small?
No. But
is it okay? So it's okay, right?
Not yet open your presentation, Dr. Hindwang.
Okay. Yeah, give me a sec. Okay, so for some reason, I don't know when I open, it's a full screen. So is it okay like this?
Oh.
Huh?
You cannot present more maximize?
Yeah, I cannot. Give me a sec.
Maybe you can click present now.
Yes, I know, but I only have a 2 slides, so I don't know how to end one of the display. You know what I mean?
Maybe the committee can help your present your PPT.
I think it should be fine. Yeah, I think should be fine. So let me share it again. Is it better now? Yeah, full screen now? What?
Okay.
Okay. Or maybe the other.
Cannot the PPT, just blank.
Okay, so maybe this one. How about this?
Okay. Yes, yeah. Already.
Yes. Yeah.
Okay. Yes.
So I shared the wrong screen. Yeah, I should share the other one. Okay, no worries. So.
Okay, that's fine.
All right, so I have to say that I have to leave early. Like I could not attend the whole seminar today, and because I have a lecture from 1 to 5 in Sydney. Okay, but I already informed the students, so I could be a little bit late, like 10 minutes late or so. All right. So I
It's okay.
yeah, because I have teaching today. It's quite packed for me today.
Yes.
Okay, so my research focus on nanotechnology in the form of nanoparticles to improve the current treatment for hard to treat diseases, including cancer, antibiotic resistant infection. So my research is all about nanoparticle for biomedical applications. All right. Um, so we working with different type of nanoparticles. We working with soft nanoparticle, which are made by organic materials like polymeric nanoparticle. So we synthesize nanoparticle with different shapes, as you can see from here, like spherical, rod-like, worm-like, and vesicle. We also synthesize like more complex nanostructure like a hyperbranch polymer or star polymer, okay? And I'm also working with hard nanoparticle. What are hard nanoparticle? These class of nanoparticles are important class of nanostructures, the comprising of inorganic materials such as like metals, metal oxide, silica, rare earth, and semiconductors. So these hard nanoparticles possess very unique magnetic, optical, and plasmonic properties. Therefore, they could be useful in different applications. For example, you guys know about the magnetic iron oxide nanoparticles because they are widely used as a contrast agent used in magnetic resonance imaging, MRI. So when these nanoparticles are subjected to an alternating magnetic field, these nanoparticles can produce heat, right? That's why these nanoparticles could be very useful for like theranostic application, like we can use for diagnosis and we can use for treatment of cancer, for example. And gold nanoparticle, we also synthesize gold nanoparticle with different shapes, spherical, rod-like, or star shape. Gold nanoparticle can absorb near infrared light, like a rod-like and star shape. And they can convert it to heat as well. So gold nanoparticle also can be used for photothermal therapy, for cancer therapy, and they also can be used for CT imaging, right? And the other class of nanoparticle, I would like to also want to share some of my research on this topic today is upconversion nanoparticles. They are made of rare earth elements, and they have a very unique optical property too, which could be very useful for biomedical applications, okay? So during my PhD, I developed like 4 different nanoparticle systems. As you can see here, I published 4 papers during my PhD. So one example is I made nanoparticle for cisplatin delivery, right? So in this case, here is the structure of the cisplatin, platinum 2. We modified to platinum 4, and then we made a polymer like this one is a diblock copolymer. We contain a functional group. In this case is isocyanate. So we modify our drug with the diamine functionalities here. So then this drug will conjugate to in the core of the nanoparticle and form it acts as a like pro-drug and also as a cross-linker. So you can see here it will form this cross-link, very stable, very well dispersed in water. And then when let's say in terms of application, in the presence of like reductant or reducing agent like ascorbic acid, glutathione, then this compound will be reduced to platinum 2, which is the cisplatin, and the whole structure will be disrupted. And then on the polymer chain, this unimer form will be excreted out from the body, and cisplatin will kill the cancer cell. So you can see here from the data in this paper I published in the Biomacromolecules. So you see block copolymer is not toxic with the cells. But when we compare with the free cisplatin, we can see that our encapsulated, our conjugated drug have a quite similar anti-cancer activity to the anti-cancer cell. So in this scenario, you can see we can use the drug as a prodrug, and then it can be conjugated to the nanoparticle. So this is a very simple example for you to see how we can use nanoparticle for the drug delivery. So this project I did during my PhD. Then after I finished my PhD in 2010, I joined Australian Centre for Nanomedicine. This center was established by the University of New South Wales in 2010. So right at the time that I just graduated my PhD. So this center, in this center, we continue to focus on nano and medical research, but particularly when I work during this time as a postdoc, I developed intensive knowledge in understanding the interface between the nanotechnology and biomedical applications. So during this time, we published quite a number of papers, and we filed 2 patents, and I have supervised and co-supervised a number of students during my postdoc. So as I mentioned, we use nanoparticle for drug delivery. So one of the drugs that we like to use is the doxorubicin, because doxorubicin has a wide range like activity for different type of cancer. So you can see this compound is intrinsically fluorescent, so very easy to track this molecule for cell uptake. However, the problem with doxorubicin, which is a drug working by intercalating with DNA, this drug can cause many adverse side effects, which cause a lot of distress for the patients. So that's why we aim to use a nanoparticle to improve the current treatment using doxorubicin. Doxorubicin is clinical use, right? So we want to use nanoparticle to improve the current treatment. So in principle, we would like to make a nanoparticle which contain the functional group. So doxorubicin has an amine group in the chemical structure. So we would like to use this aldehyde functionalities to conjugate with the doxorubicin using a chemical bonding. And so, you know, this ship base or imine linkage here is unstable in a mildly acidic condition like a pH 5, which represent the pH in the cancer cell, right? Or in the lysosome or endosome. That's why the drug will stay intact in the nanoparticles. In the bloodstream, and then it only can release the drug when it reaches a tumor or in the area which has a low pH. That's mildly acidic pH, environment. So in the paper we published in ACS Nano, we use the iron oxide nanoparticles. So we developed this polymer using advanced polymerization technique called RAF. I'm not sure if you're familiar, but we in Center of Advanced Macromolecule Design, where I did my postdoc, we have expertise on this technique, so we can make like the copolymer with the predetermined molecular weight. We can control the size of the polymer. So you can see here, so we have like a different functionality in this copolymer. So first, the blue one is a pack-like polymer to introduce the biocompatibility and also the hydrophilicity property for the polymer. And we have an aldehyde group here to attach with the doxorubicin, as I mentioned to you before. And the end functional group here, which is a phosphonic acid, right? Then we can use it for like as an anchoring point for the for the inosine nanoparticles. So you can see here, after the reaction, we mix the polymer and the inosine together, then this group will be attached on the surface of the inosine nanoparticle, and then the nanoparticle will carry the doxorubicin and also can disperse very well in water because we have the PEG-like polymer on the surface. So the use of nanoparticle for drug delivery has been intensively studied, right? You can find like thousands of paper in the literature about this topic. So many research groups put a lot of effort in understanding the mechanism uptake of nanoparticle into the cells, including like clustering mediated endocytosis, caviola, and so on. However, the understanding about the fate of the nanoparticles after being uptaken by the cells still very limited, right? So we went to a conference, and someone who is really famous in the field asked us this question. So how do you know that the drug will release from the nanoparticle in the cell? So you have to prove to us that you, in your system, the drug will be released from the nanoparticle system, because we done the release study, but as you can see from the literature, we always like did it in the like a buffer solution, so it doesn't really represent the real condition in like in the human body. So how we can prove that is actually is very difficult question, because if you look at the emission profile of the doxorubicin and the conjugate doxorubicin is very similar. And in the confocal image, you can see it's all red. You cannot really distinguish where is the free doxorubicin and where is the bound doxorubicin with nanoparticle, right? So it's really hard question. It's not easy to prove this. So we kept thinking and we we need to find the way how we can distinguish these two guys, like free doxorubicin and the conjugated doxorubicin. So luckily, we thought about one parameter, which is the fluorescent lifetime. So you all know fluorescent lifetime is the average time of the molecule stay in its excited state before it emitted a photon, right? So we predict that when the doxorubicin has a lifetime, so in this case, a 1 nanosecond. So when it absorbs the light, then it will stay in the excited state for 1 nanosecond before it emitted a photon. But when it these molecules are conjugated with polymer, right? So the lifetime, we predict that it will be different. The only thing in here is different is a doxorubicin lifetime. So when it binds to the polymer, then the chemical structure of doxorubicin gets stabilized. And then also when it encapsulated in the core of the nanoparticle, then it can prevent from the other quenching molecule like a water. That's why we predict that the lifetime of the conjugated doxorubicin will be higher than the free doxorubicin. So here you can see from this data, as you can see from the previous confocal image, it's on red. You could not see like week one is a free dox, week one is the bound doxorubicin, right? So, but on the right hand side, you can see here the different color in lifetime. So here the green color is the lifetime of the green color area is a 4 nanosecond. And the blue color here in the nucleus is a 1 nanosecond. So you can see the nanoparticle carries the doxorubicin, stay in the cytoplasm. And then when it releases the doxorubicin, it will enter the nucleus and intercalate with DNA. So now we can confident to say that our nanoparticle, we are green in here, 4 nanosecond, release the doxorubicin, and doxorubicin, free doxorubicin can go to the nucleus and then intercalate with DNA and then kill the cancer cell, right? So we use this technique, which is called FLIM, fluorescent lifetime imaging microscopy. And we one of the first to use this technique to prove that the release of the drug from the nanosystem in the cells. And we use this technique for different system and also like for different research question to solve different research question, and we publish in a number of papers here. All right, so like it is well established that nanoparticle size, surface charge, and materials composition all modulate the cell uptake, right? However, what is the influence of the shape of the nanoparticle on drug delivery? So this knowledge is still is not very well understood, right, in the literature. So we are interested in to make different shapes of the nanoparticle with the same type of polymer. So in this case, we will use RAP polymerization again. So we will start up the macro RAF agent. So in this case, is a paclide polymer. And then we use another monomer, maybe styrene, or you can do different monomer. But in this case, we use styrene. And we dissolve in these two components in methanol with monomers soluble in methanol. And then we will carry out the polymerization. So over time, the yellow component in here increasing in length, right? So the yellow polymer does not soluble in methanol, which is a solvent for polymerization. That's why during the polymerization, the polymer will be, like the structure will be reorganized and will form the nanoparticles, you see? So at the beginning, it will form the micelle, the spherical nanoparticle. And then if we keep increasing the length of the hydrophobic, in this case, a yellow block, then we can obtain like rod line, worm line, and vesicle. Yeah, so by using this method, we can obtain the high concentration of the nanoparticle in one batch. So it's different from the iceberg method, like traditional method, we only can obtain a very low concentration method, low concentration of nanoparticles. All right. So then we have a different morphologies of nanoparticles. We then will treat these four nanoparticles with the cells. In this case is a breast cancer cell line, MCF7, and all these nanoparticle were conjugated with doxorubicin. So as you can see from the data we published in polymer chemistry, the nanoparticle with high aspect ratio, in this case a worm line and rod line, will have a better cell uptake. And in this case, the nanoparticle like with cylindrical shapes were 5 times more cytotoxic than the spherical nanoparticles. So we now you see that our data suggested that the nanoparticle with high aspect ratio, like a rod and worms, could be used for drug delivery application where nuclear access is advantageous, right? And spherical nanoparticle, like a micelle and vesicle, for application where the cytoplasmic localization is required. So that means in this case, the shape of the nanoparticle actually modulate the drug delivery. So at that time, we observed this phenomenon. So it was quite interesting for us, because normally when we talk about drug delivery, we always think about like we use the spherical nanoparticle, right? But in this case, the shape of the nanoparticle actually influence significantly on the drug delivery. So we don't really know how to explain the data. We only observe the data, but we don't really know how to explain, because the mapping of the molecular mobility across the cellular compartment is very difficult, because with the current imaging technology, we don't really have enough like spatial temporal resolution, right, to to follow the diffusive route of the nanoparticles. So I was lucky at that time. Then one researcher, her name is Liz Hin. She just came back from US, and she learned a technique which is called pair correlation. Um, this method is quite complicated, but she learned in US is a form of the correlation spectroscopy. So she's using this method to map the molecular mobility of polymeric nanoparticle or doxorubicin with the millisecond and submicron resolution. So you can see from her data here. So we use the micelle and vesicle, which is a spherical, and rod and worm structure, which is a cylindrical shape, right? So in the third row here, you can see when you look at micelle and vesicle, the nanoparticle cannot really cross the nucleus membrane here. So it just stay in the cytoplasm. You see some signal here, because so we think that due to the division of the cells, that's why it carries a signal. But you see the signal cannot cross the nucleus membrane. It's very similar to the vesicle. But very interesting for the rod and the worm light. So you can see, so the nanoparticle can cross the nucleus membrane in this case. Yeah, that's why in the nucleus, you will have more concentration of the doxorubicin. That's why the nanoparticle will be more cytotoxic with the cells. All right. So it's really powerful technique. So we can use to map the diffusive route of nanoparticles. So we published this paper in Nature Nanotechnology. So it's quite interesting work if you would like to have a look. So the second topic I would like to share with you today is we use a nanoparticle for the delivery of very special molecules, which is a nitric oxide. So nitric oxide is an important cellular signaling molecule, right? This molecule is produced endogenously in our body, right? So it's a very important molecule in our body. You see how it's important when it was named as a molecule of the year in 1992, and this molecule is a subject for Nobel Prize in 1998. So in some scenarios, the underproduction of nitric oxide due to the like a lack of exercise, lack of the healthy diet, and so on. Also by age, then the underproduction of the nitric oxide can cause like different diseases. So we need to deliver more nitric oxide to the body. But the problem of nitric oxide is this molecule is highly reactive and has a very short half-life. That is why scientists was trying to develop a molecule we can release NO in situ, right? It's very difficult to deliver the NO gas, but we can use a donor, so we can release the NO, right? So there are different classes of NO donors. So like this one is the diazonium diolate, and this one is S-nitrosothione. However, these molecules still not good enough because they still release NO, but lack of stability and specificity, right? So like these molecules, the half-life is only about half an hour, right? And also this molecule also can cause many side effects for prolonged use for patients. So our question is, we wonder if nanoparticle could be a vehicle for nitric oxide delivery. So we aim to make nanoparticles which contain the nitric oxide donor inside the core of the nanoparticle. Then we hope we can enhance the stability of the anode donor in the nanoparticles. So one example is we make the nanoparticle from like PEG and also this VDM, this one another activated ester monomer. So in this case we use we use S-nitrosoglutathione. So we can use this amine group attached on the polymer chain, and we can make these nanoparticles, right, by using the self-assembly method. And you can see from TM here is a nanoparticle, and then when we have an anode, this is a nanoparticle only polymer, and this one with the S-nitrosothione. So in the presence of the beam from the TM, the anode release, so you can see structure, it look like a hollow here. And the stability of the anode donor increased significantly. So here is a nanoparticle encapsulated with anode donor. So you can see the amounts of anode release much slower compared to the free anode donor. So to be able to use these nanoparticle for biomedical applications, so we first need to prove that we have anode release in the cells. So in this case, we use the probe, which is a DAFFMDA. So when the cell uptake this probe, then the enzyme esterase will cleave the methyl groups to release the DAFFM, this one in the cell, which is a weakly fluorescent. And then in the presence of anode, then the anode will react with the DAFFM with the probe to produce the green fluorescent emission, this molecule. So we prove that we have an anode released in the cells, okay? And then we will use this nanoparticle to treat the cells in the presence of cisplatin to see if is any synergistic effect between the anode and the cisplatin. So we tested on MRC5, which is a normal cell line, and then the BE2C, which is a neuroblastoma cells. So as you can see from this table, in the presence of anode and cisplatin, then the IC50 for the cancer cell about 5 times less than when we than the IC50 when we treat the cell with only cisplatin. And interestingly, for the MRC5, the IC50 didn't change very much. So actually, the combined formulation doesn't cause like a more toxicity to the normal cells, which is what we want, right? And we also use gold nanoparticle with nitric oxide to see if we can use the nitric oxide to kill the cancer cells. So in this case, we use a diazonium diolate. So we purge secondary amine group on the polymer chain with nitric oxide, and it formed the diazonium diolate. So here's a nanoparticle. So we have a gold nanoparticle, and on the green dot is anode donor. And we can see from this data, so when we have a nitric oxide in the particle, then the nanoparticle is quite toxic to the cell. So it can kill quite a lot of cells here at the concentration of 100 micromolar, okay? So that means nitric oxide also can kill the cancer cells. So that is another work that we done. So we also use nitric oxide nanoparticle to solve another problem, which is antimicrobial resistant infection. So you know this is a big problem nowadays. So up to 80% of chronic infections have been found to be biofilm related, right? So bacteria, they are very clever. They are embedded in this polymeric matrix, right? So when they embedded in this biofilm, then they are up to 1,000 times more resistant to antimicrobial agents. So like here in the cartoon, you can see here, so here's a bacteria embedded in this biofilm membrane, right? So then when you treat this cell with antibiotic, is the antibiotic cannot really penetrate through this biofilm. That's why the antibiotic resistance occur, right? So we in the literature, it showed that where the exposure to NO, the biofilm to the NO at the picomole or nanomole range, very, very, very small, very low range, can induce a dispersal of the biofilm. So if we have an NO in the system, then NO is signaling molecule. It could tell to the bacteria to disperse this biofilm. Okay, so this.
Sorry, Dr. Duang.
Yeah.
5 minutes left.
Yeah. Okay. No
Yes.
problem. So then we will can disperse the biofilm in the presence of the antibiotic. Then bacteria will be killed, right? So we have one example here. So we make a nanoparticle and then we attach with the xanthomycin. We use clinical use antibiotics. In the structure, antibiotic has secondary amine. That's why we can attach diazonium diolate in the nanoparticle, right? So from the results, you can see, so in the formulation with the nitric oxide, then the biofilm can disperse quickly, very significantly, like you can see here from the crystal violet assay and also the viability assay. So when you attach the nitric oxide with antibiotic, the activity, it will be much higher. We published this paper in Chemical Science. So the last topic I would like to briefly discuss with you is about the upconversion nanoparticle, as I mentioned earlier. So these nanoparticle are able to absorb the low energy photon, like a near IR photon, and then convert them into the high energy. So it's very useful for imaging, right? Because the emission is in the visible range. So these nanoparticles have very unique properties. That's why it have many applications ranging from like security, like preventing forgery, or also biomedical applications. So when you want to use this upconversion for biomedical application, it's very important to convert the hydrophobic surface, because after the synthesis, these nanoparticles are kept with hydrophobic molecule, which is oleic acid. So we have a different method here, but we chose a ligand exchange method. So we want to have a systematic study about using a different polymer, like with different anchoring group. In this case is like phosphate, sulfonic acid, or carboxylic acid, and see which one show the better performance. So we make our polymer using RAF, like I mentioned before. And then we use nanoparticle with the size is around 20 nanometer. So you see the green fluorescent because we dope with the erbium. So it shows like green emission. So from the FTIR results, so you can see here with phosphate anchoring group, we have 100% of the replacement of the oleic acid, where in contrast for the carboxylic acid and sulfonic acid, we still have some oleic acid left. That's why from the DOS data, you see it's very stable structure with phosphate, and it's not very stable with the other two anchoring group. And we also want to use simulation to check like like to support our data. So you can see here from the simulation data, the absorption energy for phosphate is the highest, right? So that's why we have the phosphate system is the most stable in comparison with the carboxylic and sulfonic acid. So that is all for my talk today. I would like to thank my collaborator and my student, and thank you for your listening. And I'm happy to take questions.
Okay. Okay, thank you, Dr. Hin Duang, for very interesting and informative presentation. Now we will begin the discussion. Are you okay with the time? No.
Yes. So I have a time for 2 questions because my class actually start at 1 p.m. Yeah, but it's fine. I already informed the student I will be late.
Okay.
So maybe 2 questions.
Maybe 2 questions.
Yeah.
While we were waiting question from participant, I have already some question for you.
Yeah.
I know that to make nanoparticle is not easy, but you can do it.
Yeah.
I want to know what the direct effect for doxorubicin nanoparticle. To more safety or to more minimum dose to use application.
Yeah, so when you encapsulate doxorubicin into the nanoparticles, so we hope we can deliver doxorubicin like in the to the target site, you know. So now when you use the doxorubicin, it will go everywhere in the body. But with the nanoparticle, like with the EPI effect, as you know, the cancer cell membrane is more leaky. So the nanoparticle maybe could like it accumulate more in the cancer cell rather than in the normal cells. So it will we hope it will increase the efficacy because of this.
Oh, okay. Okay. Thank you for your answer.
Yeah.
And I will invite two participants to give some question, maybe one question. Maybe Dr. Ian Sopian is research about drug delivery system in our faculty.
Okay, thanks for opportunity. I have one question for you. Stability problem, I think is rise in the making preparation, and I think stability problem will arise when we make nano preparation. Maybe you can share how to overcome this problem. For example, to preventing particle enriched or forming a crystal. Thank you.
So stability, can you repeat the question again?
Okay.
That's
Can you
in short, yeah. So what you want to know about the stability of the nanoparticle?
Yes, stability.
Yes,
stability. And you can share how about to overcome this.
What do you mean the stability? What do you mean about the stability?
Maybe you can share about preventing particle to be large and forming crystal.
Forming crystal?
Crystallization forming when...
Yeah, so for us, we use the polymer, which is a pack line and very short hydrophobic. So we never seen like it's very stable in water. Yeah, so it depends on what polymer you choose, you know. But for us, we normally use the outer layer. We use the pack line polymer, and then it depends on what functionality that we need, like aldehyde or like carboxylic acid. We would choose the appropriate second block. Then yeah, for us, we have to maintain the hydrophilicity of the polymer, so it will be stable. Even when after coating with the inorganic nanoparticle, we have to make sure that it well dispersed in water. All right.
Okay, clear.
Yeah. Okay, so if you have more questions, just send me an email because now I have to go to the class. Okay.
Okay.
Yeah. So I will stop sharing. So thank you so much for organizing. Okay. Bye. Bye everyone.
Bye. Bye, Dr. Hinduang. Okay. Okay, thank you for Dr. Hinduang for answering the question and for the great presentation. Maybe next I will give the time to MC, and thank you to all participants for your participation. Assalamualaikum warahmatullahi wabarakatuh.
Thank you very much for Dr. Hien Duong and Dr. Ida Musfiro. Distinguished guests, ladies and gentlemen, the next agenda is the poster session. The poster session will be held at this online room. After poster session, the event will be continued to the plenary session. Thank you. Ladies and gentlemen, before we move to our next agenda, I would like to remind you to fill the attendance of the fourth ISPST 2020 on the link which shared via group chat Zoom. Thank you. The next agenda is plenary session from Professor Mary Jo Anati Corpus, PhD, Mr. Raymond R. Chandrawinata, PhD, and Dr. Sri Widodo. That will be led by Dr. Ian Sopian. And before that, I will read the curriculum vitae of our moderator. Dr. Ian Sopian finished his doctoral degree from Universitas Gadjah Mada, Yogyakarta. His main research interests are solubility enhancement of drug through solid phase modification. One of his papers is solubility improvement of sulfate prepared with various polyethylene glycol density and different kind of hydroxypropyl cellulose by solvent evaporation method. With all due respect, we now give the time to Dr. Ian Sopian.
Thank you, Master Ceremony. Good afternoon, everyone. We are very happy to meet you at for Space Day today. Now we came to a second plenary session. In session, we have three invited speakers who have joined this session, I think. They are Prof. Mary Joan, the Corpus from Philippines, and then Mr. Raymond R. Chandrawinata, PhD from Dexa Group, Indonesia. And the last one is Dr. Sri Widodo from Faculty of Pharmacy Universitas Padjadjaran. Okay, and now we come to a presentation session. The first presentation will be delivered by Prof. Mary Joan. Before she delivers the presentation, I will read the curriculum vitae of Prof. Mary Joan first. Okay, name Prof. Mary Joan, Corpus PhD. Prof. Mary Joan finished Doctor of Philosophy in Pharmacy from University of Santo Tomas, Manila, Philippines, in 2012. She is a member of Philippine Pharmaceutical Association, or many organizations, I think. Philippine Association of College of Pharmacy, Natural Product Society of Philippines, National Research Council of the Philippines. Yes, many organizations. Her research interest in product reformulation and formulation of natural product, preclinical studies of formulated drug and natural product, and cancer chemotherapeutic natural product in vitro and in vivo. Prof. Mary has published many journal article in high impact journal. Okay. To Prof. Mary Joan, time is yours. Your time to present is 30 minutes. I will remind you when presentation time is 5 minutes. Okay, Prof. Mary Joan, time is yours.
Thank you, Professor Sopian.
Okay, thank you.
Allow me to share my screen, please. Is my audio clear?
Your voice is clear, but your slide is not yet shown.
Is my audio clear now?
Your slide is not yet
Yes.
visible. Your voice is clear.
I'm sharing my screen now. Is it visible with you, the screen?
Okay, clear.
Thank you very much. Good morning, everyone. Before I start, I would like to thank Professor Nai for inviting me to the fourth ISPST. If not with the pandemic, this should have been my second visit to Indonesia. I first visited Indonesia in 2016 when I was sent by our university for training for the ASEAN Quality Network Quality Assurance. but hoping that on the 5th, maybe I can visit already Indonesia and join you. My talk is about natural products for cancer chemo prevention. But before I start, allow me first to show you the university where I came from. I came from the University of Santo Tomas, Philippines. This is our main building. This building houses the major offices in the university, as well as the Faculty of Pharmacy, where I belong, and also the College of Science. The other building that you will see here is the building for the graduate school, which also houses the Thomas Aquinas Research Center. This is where you will see our laboratory. And the Thomas Aquinas Research Center is divided into two: researches for social sciences as well as for the natural and applied sciences. Yesterday, one of the speakers, Professor Ponchai, I still remember him because he already visited UST when we invited him as the speaker. To discuss further, when we say cancer chemo prevention, we are referring to the use of natural, synthetic, or biological chemical agents to reverse, suppress, or prevent carcinogenic progression to invasive cancer. We have 3 stages for cancer chemo prevention. This happens when the normal cell undergo initiation, promotion, and progression stage. The biological agents or biological effect of natural products may attack any of these stages, either in the initiation stage, promotion stage, or on the progression stage process. In the development of a cancer chemopreventive drug, it is important that the compound or the natural product do possess antioxidant activity, because these antioxidants, as we all know, are molecules which can safely interact with free radicals before vital molecules can be damaged. Now, these free radicals are atoms or group of atoms with odd or unpaired number of electrons that can form when oxygen reacts with certain species called reactive oxygen species. The diagram that I will show you on the next slide would explain further how these free radicals can affect and cause some negative effects into the body of humans. We have different types of reactive oxygen species. It can either be the singlet oxygen, the hydroxyl group or the hydroxyl ion, the superoxide, and the hydrogen peroxide. This may be produced at the mitochondrial respiratory chain by the activation of the leukocytes, exposure to indoor and outdoor air, and even UV light. Without the antioxidants which would act on this reactive oxygen species, it is expected that it would produce oxidative damage, such as damage on the DNA, proteins, and lipids. This will now cause causal contributions. Specifically, it may contribute to occurrence of aging, heart disease, respiratory distress, inflammation, as well as cancer. We always associate inflammation and cancer because cancer usually starts with an inflammation. I have listed on the succeeding slides natural products that have been proven and are being worked out for the chemopreventive properties. With this, these natural products will have any of the following mechanisms that may either block or even suppress the occurrence of tumor or cancer. Like, for example, ability to scavenge the free radical and produce an antioxidant activity may be associated to blocking agents, while cell proliferation and apoptosis in alike can be associated to tumor suppression or suppression of cancer. Other mechanisms also are found here, such as induction of phase 2, drug metabolizing enzymes, both phase 1 and phase 2, and even induction of DNA repair, blockade of carcinogen uptake. Similar true with tumor suppressing agents, alteration of the genes can also contribute with this, as well as other inductions such as induction of terminal differentiation and modulation of signal transduction. Vinca alkaloids. We are associating alkaloids as a toxic substance, but it is known to produce an effect or a mechanism of action that may disrupt the microtubules, causing the arrest of cells at the metaphase, which may lead now to apoptotic cell death. Looking at the previous slide that I have shown you, apoptosis or induction of apoptosis may cause tumor suppression or may produce a tumor suppressing effect. Other synthetic analogs also of vinca alkaloids produce the same mechanism of action. Thus, it is being used now as a substance with potential chemopreventive properties, such as vinorelbine, vindicine, and vinflunine. Another is phodophyllotoxin. Phodophyllotoxin can reversibly bind to tubulin. We have here the structure of etoposide, which can inhibit topoisomerase 2, thus inducing topoisomerase 2 mediated DNA cleavage, giving its effect to be also a potential chemopreventive agent. We all know paclitaxel, and this is already available in the market. It belonged to taxanes, and these drugs, docetaxel and paclitaxel, are now being used for the treatment of breast cancer. The mechanism of action of taxels and paclitaxel and other taxanes is to promote polymerization of tubulin heterodimers and microtubules, thus suppressing the dynamic changes in the microtubules, resulting in mitotic arrest. Camptothecins also, which can prevent DNA relegation and causing DNA damage, which results to apoptosis. But when it comes to different literatures for chemopreventive agents, what would be highlighted more will be the use of natural resources from marine source. This includes marine algae, coral reefs, marine herbs, marine fungi, bacteria, and seaweeds. I highlighted the most common marine sources and gave examples of those which are already identified to possess chemopreventive effect. Marine algae, knowing that they possess several essential nutrients such as lipids, minerals, proteins, fibers, fatty acids, and polysaccharides, specifically these polysaccharides, the sulfated polysaccharides, gave the marine algae a potent antioxidant effect due to its ability to react with oxygen species or scavenge oxidative stress. And this gives it responsibility for carcinogenic property. Other compounds which are isolated from marine algae are nitrogen-containing heterocycles and also sulfated polysaccharides. Second are the coral reefs. Coral reefs are already reported to have anti-inflammatory, anti-cancer, and antioxidant activities. And in the market, we already have Cytosar. This is the first marine anti-cancer drug found in coral reefs that are used for the treatment of leukemia and lymphoma. This acts by disrupting DNA synthesis in cancer cells. Other compounds isolated aside from Cytosar are dolastatin, nitrogenous diterpenes, and also sterols. These are being worked out now for chemopreventive properties. For marine herbs, there are alkaloids isolated for marine herbs. Sorbisilactone A and that of B are proven to be used for the treatment of leukemia cancer cell. These are the other compounds which are isolated from marine herbs, which are known to possess chemopreventive properties. Highlighted here is the presence of fucoidan, which is already proven for its chemopreventive properties. We also have marine fungi, which exhibits cytotoxicity against A549 or adenocarcinoma and HCT116 or the colon cancer, referring to the human colon cancer cells, and HepG2 for the liver cancer cells. Cytotoxic effect against the following cells are seen through the isolates from marine fungi, specifically viruloid. And other compounds isolated from marine fungi are as follows. The last one are the seaweeds. Seaweeds, aside from the fact that it is being used as an excipient, it also possesses several therapeutic potentials, such as the sulfated polysaccharides, which are also found on other marine sources, polyphenolic, terpenoids, flavonoids, and lipids. This particular natural source from marine origin may exhibit antioxidant effect and also anti-mutagenic activity. Specifically, we have different types of seaweeds. We have the brown, red, and the green groups. All of these classified seaweeds possess an anti-proliferative activity against various malignancy cell lines. And aside from that, the polysaccharides found on these seaweeds are known to cause dose-dependent scavenging against free radicals, thus displaying an antioxidant effect. The antioxidant effect that these particular seaweeds produce can now be used for the prevention of the proliferation of cancer cells. Other compounds isolated from seaweeds are as follows. I also have here natural products derived which are currently under clinical use. We are familiar with tamoxifen, which is a treatment for breast cancer, as well as raloxifene, which is also for breast cancer, letrozole, and finasteride, which is being used for the treatment of prostate cancer. Others is aspirin for colon cancer and celecoxib, also for colon cancer. While natural products derived from which are currently being used for clinical trials are as follows. They are looking at the following compounds, which can be a potential chemopreventive drug, such as beta-carotene, lycopene, curcumin, anthocyanin, and epicatecin. With all this in mind, knowing that the Philippines is surrounded by bodies of water, we have come up with this slide to look for a certain seaweed or a marine source which can be useful for the treatment of cancer or which has a chemopreventive effect. The seaweed that we use was collected from Batangas. It belongs to the family of Sargaceae, genus Sargassum, with a binomial name of Sargassum silicosum J.G. Augert. This is found in both northern Philippines and also on central Visayas and Mindanao. Based on the literature of the different species of Sargassum, Sargassum species contains strong antioxidant activity and anti-tumor and chemopreventive properties. Thus, since there are no studies yet when it comes to the Sargassum which is being found in the Philippines, we push through with determining the effect of this Sargassum species, the Sargassum silicosum, to further check it for its chemopreventive properties. Aside from the fact that it has those particular biological effects, it is also found that different species of Sargassum contains polysaccharides, polyphenols, fucoidants, and polyphenolic compounds. If you will recall on the previous slides, these are the constituents which are being highlighted to possess chemopreventive properties. And other species of Sargassum, such as alginate, fucoxanthin, phyophytene, and monosaccharides. With this in mind, we came up to determine the antioxidant and chemopreventive properties of Sargassum, as well as to determine its anti-initiating activity and anti-promoting and anti-progression activity using several tests. For the methods, after the collection of and extraction of the plant, we initially test it for the presence of phenol and flavonoid. and further test the one with the highest amount of phenolic and flavonoid content. We further test it for its chemopreventive properties. For the chemopreventive properties, as what I've mentioned, we based it on the stages of cancer chemoprevention cascades: anti-initiating, anti-promoting, and anti-progression activity. We use solvent partition for the extraction of the plant sample, which yielded 4 different fractions: water butanol, water methanol, water fraction, and hexane fraction. These are the biological tests we performed to objectives. Preliminarily, we tested it for total phenolic content and total flavonoid content. For the total phenolic content, we use Folin Schuckel 2 solution and gallic acid as the standard. For total flavonoid content, we use aluminum chloride as the reagent and quercetin as the standard. To whatever is the highest when it comes to TPC and TFC, or total phenolic and total flavonoid content, we further test it for its scavenging activity using hydroxyl radical, nitric oxide, and hydrogen peroxide. We have selected these three because specifically nitric oxide is associated with inflammation. And we are looking forward to developing not just determining the chemopreventive properties of the extract, but also further developing it as an anti-inflammatory agent and even linking it for allergy or anti-allergy drug, anti-allergy agent. If not with the pandemic, I should have shared with you our results for the anti-allergy research. But unfortunately, we have to repeat some of the procedure because we closed down during the pandemic. So for the scavenging activity, for the hydroxyl radical, we use the positive control ascorbic acid and the following reaction mixture, which makes use of ferric chloride EDTA and that of hydrogen peroxide. This is based on the ferric oxide EDTA and hydrogen peroxide reaction, what we tend to call as the Fenton reaction. The reaction mixture is composed of the following. Next is nitric oxide scavenging. Nitric oxide scavenging activity is based on the reaction mixture, which consists of sodium nitroprusside. We also make use as ascorbic acid as the positive control or the standard. For hydrogen peroxide, of course, we use hydrogen peroxide sample mixture and also ascorbic acid as the standard. After which, we determine the percent scavenging activity of the three scavengers: hydrogen peroxide, hydroxyl, and nitric oxide. For the cell culture preparation, we make use of UVM1 or melanoma cancer cell line because we are trying to develop here a possible topical preparation. So this UVM1 was obtained with ITCC number. It was incubated for 24 hours initially, 5% carbon dioxide with 37 degrees centigrade temperature. We also make use of normal cell line, particularly collecting it from human foreskin, just to compare specifically if the sample is toxic to normal cell. The same condition, aside from the fact that it is cultured using RPM-I, it was also maintained under carbon dioxide 5% and incubated at 37 degrees Celsius. For anti-promoting activity, we use trypan blue test or dye exclusion test for cytotoxicity test by MTT assay. For trypan blue test, the cells were suspended, and then after which When the cells are already seeded per well, the extract was added and then trypsinized. After which it was centrifugated, and the total number of cells and stained cells were counted under the microscope using hemacytometer. The cell density is what we are looking at for trypan blue test. For the cytotoxicity assay, of course, we use MTT assay, wherein the cells were incubated here for 24 hours, after which different concentrations of the extracts were added, and then we allowed the plates to proliferate. Plates containing the cell, we allowed them to proliferate, and then we added the MTT reagent, after which it was incubated at 37 degrees for 4 hours. Cells were removed using the MSO, and then the absorbance was measured at 500 nanometer. In here, percent cell inhibition was measured. For anti-progression activity, apoptosis assay was used. For the apoptosis assay, 96 well plates were used to seed the cells, and then after that, we allowed the cells to equilibrate at room temperature. And then caspase glow 37 was used, after which the contents were mixed using a plate shaker and then incubated for 30 minutes, 1 hour, and 2 hours. The luminescence was measured here using Promega luminometer. For the results and discussion, based on the results for the total phenolic and total flavonoid content of the different fractions, it shows that 3 extracts, DCM, methanol, and methanol, water, and hexane, gave the highest gallic acid equivalent and quercetin equivalent. These were tested further for its scavenging activity. For the hydroxyl radical scavenging activity, we tried to compare this with ascorbic acid, and it shows that the highest scavenging activity or highest inhibition was seen with the extract DCM. Similar with the other scavengers, nitric oxide, also DCM showed a potential increase in the percent inhibition, comparing it with that of ascorbic acid. Same as true with hydrogen peroxide. Also, DCM gave the highest scavenging activity. As a summary, if you will look at the inhibitory concentrations of the fractions of the extract as compared to that of ascorbic acid, the one with the highest inhibitory concentration as compared to ascorbic acid is dichloromethane or DCM in all of the different scavengers, hydroxyl, nitric oxide, and hydrogen peroxide. For trypan blue test, we try to compare our extract with an anti-cancer drug, mitomycin. And based on the results, it showed that the effect of the extract as well as that of the anti-cancer drug mitomycin is dose-dependent. It is seen visibly on the extract, sargassum silicosum extract, which is shown here in yellow, that it is dose-dependent. As we increase the concentration of the extract, the viability of the cells decreases. For the results for the MTT assay, which will tend to show the percent growth inhibition of the cells, it is also dose-dependent. UVM1 cells treated with the extract show that increasing concentrations would also increase the percent inhibition. The 50% inhibition of the cells for UVM1 cells treated with the extract is seen to be within 50th concentration 70. In between this graph, 50 to 100, particularly 70.18 micrograms per ml. While for the UVM1,
time presentation, 5 minutes left.
Thank you, sir. For UVM1 treated with mitomycin, this is our anti-cancer drug. It kind of show an erratic value, maybe because of its concentration. This is a concentrated drug and it's potent. But comparing it with the results of that of the extract, still mitomycin has a powerful percent growth inhibition as compared with our extract. With the percent inhibition on normal epithelial cells when treated with the extract, it also showed a dose-dependent manner when it comes to the response with cell inhibition or growth inhibition. For the apoptosis assay, we base it on the luminescence, comparing the effect on the 30th minutes, 30, 60, and 120 minutes. And based on the results, UVM1 treated with the extract also showed a dose-dependent effect with a maximum of luminescence seen at the highest concentration. For the one treated with mitomycin, the concentration also showed a dose-dependent manner for both 30, 60, and 120 minutes. And a lesser luminescence appeared for normal epithelial cells treated with the extract. For the conclusion, based on our research, dichloromethane hexane and methanol water extract of S. silicosum shows a strong antioxidant activity owing presence of polyphenolic compounds. The dichloromethane showed IC50 with the lowest in all of the antioxidant assays, which supports the use of Sargassum silicosum as potential scavenger for free radicals that may prevent the initiation of cancer cells. While the extract also shows strong cytotoxic effect on UVM1 cancer cells or melanoma cancer cells, which were reported to have an IC50 of 70.18 after 24 hours exposure, as compared to mitomycin, which has an IC50 of 196.4 micrograms per ml. And also a low cytotoxic effect for normal epithelial cells with IC50 of 1,174 micrograms per ml. The mean luminescence of the normal cells is significantly less with mitomycin and the extract. And mean luminescence using mitomycin C is significantly less than that of the extract. And these findings indicates that the extract is more effective than mitomycin C when it comes to luminescence. We are recommending that we can also test this extract for other cell lines. I would like to acknowledge the following for the success of this research, including my co-collaborators. Thank you.
Thank you for presentation. Very interesting and informative presentation, I think. Maybe there will be a lot of questions, I think, from this presentation, but we will set it first. The discussion session will begin until all speakers have been present. You can write down in chat room first. Okay, ladies and gentlemen, now we going to a second presentation. The second presentation will be delivered by Mr. Raymond Chandra. Good afternoon, Mr. Raymond. Are you ready to present?
Yeah, good afternoon, Dr. Ian. Thank you very much for inviting me.
Okay. Before you present, I will be read short curriculum vitae first. Name Raymond R. Chandra, PhD. Raymond PhD is molecular and clinical pharmacologist who work in area drug discovery. Drug discovery, I think, interest field of this. He received doctorate degree from University of California, Riverside, and underwent postdoctoral research fellowship in the Department of Medicine, University of California, San Francisco School of Medicine. I think many achievement very prestigious. They are more than 10 original Indonesian modern medicine product, amazing, that have been launched in Indonesia and abroad. He received Habibie Award, and Sinta Award. I think a prestigious achievement. And what intellectual property organization, Medal in Geneva, Switzerland. Now serving a director business development at Dexa Group. Yeah, I think very good ZP. Okay, Mr. Redmond. Now time is yours to present. Your time to present is 30 minutes. Maybe we will remain if presentation session until 5 minutes left. Okay.
Thank you very much for your kind introduction, Dr. Ian Zopian. Also, I'd like to acknowledge and my thankfulness to the invitation to Professor Utai, Professor Mutaryadi, and this very prestigious international seminar. What I will be talking about today is actually about OMI, Indonesian Modern Herbal Drugs. But allow me to speak in Indonesian because I think the majority of the participants are Indonesian, and I think it's appropriate for me to speak Indonesian this particular topic. Ya. So selamat pagi Bapak Ibu sekalian, terima kasih atas undangannya yang saya hormati sekalian. Jadi saya akan mengetengahkan suatu makalah yang bertema potensi pengembangan obat modern asli Indonesia untuk ketahanan bahan baku obat di Indonesia. Jadi Bapak Ibu sekalian, yang kita ketahui dan pasti Bapak Ibu sekalian banyak sekali melakukan penelitian, riset dan development di bidang obat herbal. Dalam hal ini kami melakukan ini juga tidak hanya pada herbal, tapi juga pada pembicara sebelumnya bicarakan mengenai marin, kemudian bakteri dan lain-lain. Jadi itu sebenarnya biodiversitas alam. Dan Indonesia ini tidak kalah dengan negara lain, bahkan kita mempunyai yang paling banyak sekali yang kalau kita mau mengeksplorasi, kita akan mendapatkan obat-obat baru daripada biodiversitas alam Indonesia. Kita selalu harus cepat di sini karena kompetitor kita di luar negeri juga melakukan dan banyak sekali yang mereka datang ke Indonesia sehingga kalau hilang nanti kita sendiri yang akan melongo karena kecolongan. Banyak sekali terjadi. Jadi Bapak Ibu sekalian, para peneliti, adik-adik farmasi dan lain-lain semuanya, para dosen, para peneliti, kita mempunyai memang lebih dari 7.000 medicinal plant yang jadi tanaman yang bisa dibuat dipakai untuk mencari obat-obatan baru. Nah, bagaimana caranya? Nah, ini yang saya akan bicarakan hari ini, apa yang kita sudah lakukan. Jadi hari ini saya bukan menggurui, saya tidak ini, tapi saya hanya sharing apa yang kami sudah lakukan, yang paling kami susah lakukan dan juga suka dukanya dan nanti saya akan ceritakan. Namun kita mesti ingat bahwa memang di dalam badan klasifikasi Badan POM ada 3, jamu, obat herbal standar, dan fitofarmaka yang tentunya adik-adik dan bapak ibu sekalian sudah tahu apa perbedaan dan jarak kita. Kalau jamu adalah terbukti secara empiris saja. Kalau obat herbal standar, nah ini kata standar penting sekali, harus ada standarisasi bahan baku maupun obat jadi. Sehingga apa yang dilakukan, kita harus melakukan standarisasi dan juga dilakukan praklinik, terutama yang dibutuhkan adalah data toksisitas dan farmakodinamika, dan akhirnya data baku mutu. Ini penting sekali dilakukan untuk memastikan bahwa obat kita adalah terstandar. Ya. Nah, apabila dilakukan uji klinik, maka kita akan mendapatkan yang disebut sebagai fitofarmaka. Nah, klasifikasi yang terbaru untuk obat herbal terstandar dan fitofarmaka sekarang kita sebut sebagai OMI. Obat modern asli Indonesia. Ya. Nah, sebelum kita melangkah ke OMI-nya, kita bicara mengenai fitofarmaka karena ini adalah tingkatan yang paling tinggi di bawah Badan POM. Harus dilakukan standarisasi, tapi sebaiknya menurut pengalaman hemat kami harus dilakukan dimulai dengan penapisan molekuler dari ekstrak maupun fraksi. Pembicara sebelumnya sudah menunjukkan bahwa beliau melakukan penapisan menggunakan enzim assay, tapi tidak hanya enzim assay, bisa juga dilakukan menggunakan berbagai macam teknik-teknik molekuler, menggunakan ELISA, menggunakan RT-PCR, menggunakan apa saja yang kita bisa kuantifikasi, asal itu bisa terstandar dan tervalidasi tentunya. Jadi ini yang kita lakukan assay clinical biology di tingkat sel maupun hewan coba. Penting sekali untuk menemukan mechanism of action dan farmakodinamika daripada obat yang kita cari. Kemudian pasti lakukan profil toksisitas komprehensif. Apa saja? Standar yang kita lakukan harusnya adalah toksisitas akut, subkronik atau kronik, genotoxicity itu pada mutasi, dan juga teratogenisitas. Ini paling enggak 4 ini yang harus kita lakukan ya. Dan akhirnya kalau fitofarmaka harus lakukan uji klinik dengan placebo atau kontrol aktif obat yang bisa kita bandingkan secara menurut CUKB, cara uji klinik yang baik atau good clinical practice ya. Jadi ini adalah standar daripada fitofarmaka yang harus kita lakukan ya. Nah, tentunya Bapak Ibu sekalian, adik-adik sekalian, farmasis, apoteker, kita tahu bahwa di dalam pembuatan proses obat herbal, tergantung dari kompleksitas dan kemurnian, kita bisa menggunakan simplicia, ya ini hanya dalam pengeringan, atau diekstrak, diekstraksi, dan difraksinasi. Otomatis ini adalah ekstraksi bertingkat. Nah, namun yang kami lakukan adalah yang disebut sebagai fraksinasi dengan screening molekuler, atau yang disebut sebagai bioactive fraction. Nah, di sini satu step lagi udah mendapatkan compound atau senyawa. Jadi ini adalah menggunakan senyawa fraksinasi, isolasi, dan sintesa apabila dibutuhkan kuantitas yang tinggi, yang banyak sekali. Yang kita banyak lakukan adalah bioactive fraction, yaitu fraksinasi dengan screening molekuler. Ya. Jadi kita bayangkan kalau misalnya di sini mungkin di simplicia ini mungkin ada 1.000 atau 2.000 senyawa di dalam satu tumbuhan, ya mungkin lebih dari itu. Nah, itu kan dibagi dari grup-grup, ada alkaloid, flavonoid, dan lain-lain. Tapi kalau sampai bioactive fraction, kita pastikan bahwa tidak hanya lebih dari 10-20 senyawa saja. Karena kalau banyak juga nanti kalau berhubungan dengan reseptor daripada di mana dia berikatan, maka kalau tidak terlalu banyak, maka ikatan reseptornya akan lebih bisa terkuantifikasi daripada kalau kita menggunakan banyak sekali senyawa yang ada di dalamnya. Ini sebenarnya mendekati, belum persis, tapi mendekati dengan suatu compound, suatu senyawa yang berikatan dengan reseptor. Nah, banyak yang bertanya kepada saya, apa sih fraksi itu? Nah, saya tunjukkan seperti berikut ya. Jadi di dalam suatu tumbuhan ada juga itu sebenarnya terbagi-bagi bagiannya, kita tahu ya. Di sini ada rizoma ya, root ya, akar ya, trunk di bagian ini kok. Paclitaxel yang tadi disebutkan itu diambilkan dari bark. Jadi itu adalah kulit kayu di sini daripada trunk. Jadi fraksi daripada trunk tersebut dan diisolasi senyawanya. Atau branches ini adalah cabang-cabang atau sampai leaf atau buahnya dan lain-lain ya. Tapi yang kami lakukan bukan hanya menggunakan salah satu daripada ini, tapi lebih lagi di fraksi lagi di sini. Kita bisa mendapatkan fraksi daripada daun, kita bisa mendapatkan fraksi dari buah, atau dari akar, atau dari kulitnya, dan sebagian besar. Jadi ini adalah fraksi-fraksi. Jadi lebih sedikit lagi, lebih kecil lagi dibanding dengan keseluruhan tanaman yang kita gunakan. Ya. Nah ini contohnya, saya memberikan contoh. Kenapa fraksi itu penting sekali? Kami melakukan salah satu ini dari berbagai macam penelitian. Ini adalah fraksinasi daripada Valeria macrocarpa atau yang kita sebut sebagai mahkota dewa. Nah, ini kita mesti bangga punya mahkota dewa ini. Karena ini adalah salah satu tumbuhan yang memang tumbuh dan berasal dari Papua, dari Irian Jaya. Nah, di antara 40 fraksi yang kami sudah lakukan terhadap Valeria macrocarpa, buahnya saya hanya menunjukkan 4 saja. Ya, jadi kami namakan ini secara chemometri, DLBS 1442, DLBS 1433, DLBS 1449, dan DLBS 1425. Dan masing-masing menduduki area di mana secara kombinasi mempunyai pattern daripada fingerprinting daripada senyawa-senyawa tertentu. Jadi 42 dengan 39 ini lain. Kalau kita lihat secara 3 dimensi, yang ini sebelah kiri atas, yang ini sebelah kanan kiri bawah. 1449 juga di kanan bawah, kalau ini lebih kanan, lebih atas ya, sama-sama di kanan. Yang selanjutnya kami tidak punya. Nah, tapi intinya demikian, setiap daripada fraksi daripada hanya buah mahkota dewa ini, kembali lagi ini hanya 4 dari 40 fraksi yang sudah kami lakukan, mempunyai sifat farmakologis dan senyawa yang berbeda-beda. Tentunya kita sudah meneliti daripada kegiatan daripada setiap senyawa di sini, tapi cukup dikatakan bahwa fraksi 1442 dengan fraksi 1433 itu mempunyai sifat farmakologis yang berbeda. 1449 dengan 1425 juga sifat farmakologis berbeda. Ini dengan ini juga beda, ini dengan ini juga beda, ini dengan ini juga beda, ini dengan ini juga beda. Jadi setiap fraksi mempunyai sifat farmakologis yang berbeda-beda. Jadi dapat dikatakan bahwa memang fraksi itu mempunyai efek terhadap gen dan protein tertentu. Kenapa dia mempunyai sifat farmakologis berbeda-beda? Contohnya 1442 ini kami sudah pasarkan sebagai dismeno. Obat dismeno ini nanti saya tunjukkan, ini adalah obat yang sekarang digunakan oleh para dokter-dokter kandungan kebidanan di seluruh Indonesia untuk mengobati endometriosis. Kemudian 1443 ini bersifat sebagai pengantar hepatoprotektor, fibrosis reversal dan lain-lain. 1449 ini lebih penaik daripada HDL, pengurangan daripada trigliserida, dan 1425 ini untuk pada kanker. Jadi kita lihat di sini, dari satu buah saja maka dewa kita bisa menggunakan fraksi yang berbeda-beda. Nah, tahunya di mana fraksi itu mempunyai sifat yang tertentu. Jadi ini kembali yang tadi menunjukkan kita harus menstandarisasi setiap fraksi, dan kalau bisa satu hari kita bisa mengisolasi dan elucidasi struktur daripada fraksi tersebut. Jadi dari salah satu fraksi ini bisa didapatkan kan di ini setiap fraksi saya katakan ada 10 sampai 20 senyawa ya. Jadi salah satu atau salah 10 bahkan bisa didapatkan senyawa dengan teknik farmakologi molekuler ya. Nah, bagaimana yang kita lakukan? Apa yang kita lakukan untuk mendapatkan sifat farmakologis? Kami menggunakan sistem yang kami sudah trademark dan patenkan, itu adalah tandem chemistry expression bioassay system. Jadi kita menggunakan sistem ini, tandem chemistry expression bioassay system, supaya lebih cepat pengembangan obat itu. Kita tahu bahwa pengembangan obat itu adalah bisa sampai 5 sampai 10 tahun. Nah, dengan ini kita bisa mendapatkan kombinasi ini bisa sampai 2 sampai 3 tahun. Namun pengembangannya dengan uji klinik dan lain-lain masih memang membutuhkan 5 sampai 6 tahun. Tapi intinya adalah TCEP adalah metode screening dipatenkan untuk mendapatkan kandidat fraksi yang baik, yang paling efektif dan aman untuk pengobatan. Nah, tahunya dari mana? Pertama, kita akan melakukan semua screening terhadap molekuler. Jadi ini kita harus tahu dengan target gen dan protein. Kita tidak pernah melakukan saya punya tanaman apa, saya mau buat apa. Secara empiris buat apa? Karena menurut saya itu untuk jamu. Tapi kalau kita membuat obat modern, kita mulai dengan penyakitnya dulu, patofisiologinya apa, gennya apa, proteinnya apa, sekarang metabolomicsnya apa yang mengandung, yang kacau di dalam penyakit itu, sehingga itu yang menjadi marker kita untuk bisa men-screening berbagai macam fraksi-fraksi yang kita dapatkan. Jadi ABCDE ini adalah fraksi-fraksi dari satu tanaman ataupun campuran tanaman atau beberapa tanaman. Sehingga kita lakukan ekstraksi bertingkat sampai akhirnya kita mendapatkan yang disebut sebagai produk atau kandidat obat, druggable material. Kemudian druggable material ini otomatis yang kita bisa isolasi karena semua akan harus di-screening terhadap target gen protein, maupun sekarang kita sudah mulai screening terhadap metabolik. Nah, DLBS potential product ini harus druggable. Artinya apabila kita menggunakan berbagai macam sistem jadi simulasi, maka dia akan mempunyai reseptor tertentu sesuai dengan target gen dan protein. Attachment-nya itu kita bisa menggunakan surface plasma resonance dan lain-lain, sehingga di dalam simulasi secara in vitro kita menentukan bahwa ini obat ini memang kandidat obat ini yang berbentuk fraksi memang punya farmakologi yang baik sekali. Dan tentunya ini kita gunakan berbagai macam expression bioassay system, bisa sel fisiologi, secara kuantifikasi di bawah mikroskop dan lain-lain, ekspresi gen, biochemical assay, analytic metabolic, yang sekarang metabolomics sudah mulai kita bermain, dan akhirnya kita harus tentukan juga animal pharmacology untuk mencari farmakodinamika secara klinis. Itu penting sekali, semua ini kita lakukan. Nah, otomatis apa yang dilakukan? Ya, kita menggunakan area gen spesifik yang kita bisa download dari NIH, National Institute of Health. Di situ NCBI banyak sekali mengandung gene protein yang sudah menjadi druggable dan sudah diakui oleh FDA, US FDA sebagai ini adalah target-target obat yang kita ketahui. Jadi mulai dari ujung kepala sampai ujung kaki, ya, ujung kuku kaki ini ada semua gennya. Kalau kita mau cari di NCBI itu semua ada gennya, tergantung daripada penyakit apa yang kita mau. Jadi Bapak Ibu sekalian, kita mulai bukan dari tanaman, kita mulai dari penyakit apa yang kita mau. Baru kita cari target gen dan proteinnya. Dan otomatis kita menggunakan modern pharmacological techniques, jadi kita menggunakan database tadi yang kita lakukan dari NCBI, kita mempunyai protein target kita dapatkan dari NCBI, kita nulis secara gratis, kita punya chemical diversity yaitu fraksi dan kita lakukan bioactive fractionation dengan menggunakan TCEP, tandem chemistry expression bioassay system, kita cari hitnya, kita harus menggunakan optimalisasi daripada heat structure, ini penggunaan daripada teknik-teknik daripada phytochemistry ataupun kimia, dan lain-lain. Kadang-kadang kita harus menggunakan berbagai macam metode untuk memperbaiki daripada kristal, daripada strukturnya. Akhirnya kita lakukan dari toxicity dan juga farmakodinamika, dan akhirnya kita lakukan di manusia, melakukan uji klinik. Jadi kalau kita lihat di sini, apa yang kita lakukan ini semua yang dilakukan oleh multinational company kita juga sudah lakukan, sehingga memastikan bahwa validasi daripada obat kita benar-benar memenuhi persyaratan internasional secara molekular farmakologi. Akhirnya juga harus dilakukan fase dari uji klinik, fase 1, 2, atau fase 3. Kalau di Indonesia tidak perlu fase 1, fase 2, langsung fase 3 boleh, asalkan kita bisa menunjukkan toksisitasnya bagus. Tapi kami tetap melakukan fase 1 karena kami memastikan bahwa pasien tersebut benar-benar aman mendapatkan fraksi atau kandidat fraksi yang kita lakukan dengan menggunakan tandem chemistry expression bioassays system tersebut. Sehingga apabila kita lakukan keamanan, baru kita berani melanjut ke fase 3. Fase 1 ini selalu di manusia yang sehat, fase 3 di manusia yang sakit. Jadi kita lakukan di fase 3 ini supaya mendapatkan hasil yang bagus, yang bisa tervalidasi secara klinis, kita lakukan dengan dokter-dokter Indonesia, di Rumah Sakit Indonesia, dengan pasien Indonesia, sehingga obat tersebut memang benar-benar tersertifikasi sebagai obat modern asli Indonesia. Nah, apakah dengan demikian klinisi sudah siap menerima obat Indonesia? Yang pasti mereka tidak mau meresepkan adalah jamu. Yang diterima adalah klasifikasi OHT dan fitofarmaka. Karena sudah ada data dan terstandarisasi, itu yang penting. Bahwa OMA itu adalah OHT dan fitofarmaka. Jadi tantangan kita saat ini adalah kita harus masih meyakinkan dokter bahwa efikasi obat herbal yang didesain secara baik, secara farmakologi modern adalah sama efikasinya dengan obat kimiawi. Nah, ini banyak dokter yang masih menyaksikan. Jadi kita perlu menggunakan edukasi dan sosialisasi bahwa obat hasil penelitian daripada peneliti Indonesia adalah sama baiknya dengan obat kimiawi. Nah, untuk itu kita memastikan bahwa dilakukan uji klinik yang baik, CUKB, GCP untuk obat-obat yang ditemukan di sini ya, dan lain-lainnya. Nah, ini masalahnya di Indonesia, penggunaan obat herbal masih minim ya. Ini tahun 2013-17 7 tahun yang lalu. Kalau ditanya sekarang pada hari ini, Hari Sumpah Muda, 28 Oktober 2020, apakah masih minim? Masih minim, saya katakan. Penggunaan obat herbal, walaupun sudah dilakukan berbagai macam penelitian, masih minim. Tapi sekarang ada sudah titik pencerahan. Ya. Malah pada para dokter masih memikirkan penggunaan obat tradisional, obat herbal masih bisa menghambat terapi. Nah, ini yang kita harus hilangkan. Di dalam stigma dokter dengan menggunakan sosialisasi, dengan menggunakan banyak sekali edukasi, kita menunjukkan bahwa obat modern asli Indonesia tidak kalah efikasinya dengan obat-obat kimiawi. Kalau enggak, mereka merasakan bisa menghambat terapi. Dan ini menjadi PR buat kita semua. Anda sebagai apoteker, Anda sebagai farmasis, juga kita semua bertanggung jawab memastikan bahwa obat herbal Indonesia yang berasal dari 7.000 tanaman Indonesia, kita tidak bisa menunjukkan, kita harus bisa menunjukkan bahwa dia tidak kalah dengan obat kimiawi. Karena kita ini sudah banyak importasi, bahan baku obat 99% itu diimpor dari seluruh dunia. Nah, kalau kita lihat, mengapa salah satu penghambatan? Karena kalau kita lihat nih, dokter-dokter yang biasa meresepkan obat herbal, di South Korea itu 15,26%, di Cina 13%, Taiwan kira-kira 10%. Lumayan, di Indonesia baru 2%. Kadang-kadang 1, berapa persen, jadi tidak banyak yang meresepkan obat Indonesia, obat hasil dari obat modern asli Indonesia ini. Dan kalau kita memeriksa, berkaca diri kepada Jerman, di Jerman hampir 50% yang bisa meresepkan obat-obat yang berasal dari naturopati, ya. Sehingga kita ini masih lebih banyak sekali, masih banyak. Jadi ini di sini nah, fitoterapi ini lebih dari 50% kalau kita lihat ini dokter-dokter di Jerman yang menggunakan fitoterapi, ya. Jadi kita masih banyak PR bagaimana mengedukasi sesama nakes, yaitu seorang dokter, mereka harus bisa meresepkan obat herbal apabila obat herbal tersebut ditemukan di negara kita. Sehingga kalau kita lihat definisi OMA yang sekarang harus kita lakukan dan harus kita panjatkan itu apa? Obat-obat dari Indonesia adalah suatu sediaan bahan alam Indonesia, bisa berupa ekstrak, tidak harus fraksi, bisa juga ekstrak, tapi harus tumbuh di Indonesia dan harus kalau bisa tanaman asli Indonesia, ya, atau pernah ditulis-tulis dalam buku herbal Indonesia. Contohnya buku cabai puyang warisan nenek moyang, atau buku tulisannya Pak Seno dan lain-lainnya. Dulu ada seorang Belanda itu yang menulis, saya lupa namanya, seorang ibu. Itu juga boleh, tapi pernah ditulis karena itu menggunakan adalah di bagian daripada jamu-jamuan dan tanaman asli. Ya. Kemudian riset penemuannya dilakukan di Indonesia serta mempunyai data. Nah, ini yang diharapkan adalah benar-benar data-data yang digali daripada penelit riset Indonesia dengan mekanisme kerja yang jelas. Mekanisme kerja yang jelas ini pastinya harus kita menggunakan teknik molekuler dan farmakodinamika di in vivo hewan coba. Diproduksi secara farmaceutica modern, contohnya apa? Bisa bentuk tablet, bisa sirup, bisa bentuk kapsul, dan lain-lain. Tapi tidak tinktur dan lain-lain ya. Ini harus diproduksi farmaceutica modern dan akhirnya mempunyai mempunyai status sebagai obat herbal standar maupun fitofarmaka dari Badan POM. Nah ini kalau ini contoh di mana banyak menteri, jadi ini yang ini Kemristek Pak Bambang ya, Prof Bambang. Kemudian di sini juga ini Pak Terawan ya, kemudian di sini Pak Gumiwang jadi seorang menteri perindustrian. Mereka semua mengendorse obat modern asli Indonesia ini. Kenapa? Jelas sekali kalau kita mengembangkan lebih banyak obat modern asli Indonesia, bisa mengurangi import bahan baku yang memang banyak sekali. Dan Bapak Terawan Agus Putranto, Dokter Jenderal Terawan Agus Putranto mengatakan bahwa memang obat modern asli Indonesia ini sudah dikembangkan, ini dari kekayaan alam Indonesia, terdapat lebih dari 80 OMA yang terproduksi di Indonesia, Stimono dan sebagainya. Dan bukan hanya harusnya dipakai di Indonesia, tapi sudah mampu diekspor ke beberapa negara Eropa dan Asia. Jadi dikatakan lebih dari 80 OMA yang sudah di Indonesia, sebetulnya banyak sekali yang bisa digunakan. Nah ini diperkuat lagi dengan suatu buku yang baru saja diterbitkan oleh Badan POM, April lalu, yaitu Informatorium Obat Modern Asli Indonesia di masa pandemi COVID-19. Jadi ini adalah publikasi Badan POM yang Anda sekalian bisa download. Di sini akses online-nya perpustakaan Badan POM. Anda bisa download PDF-nya. Ini mempunyai banyak sekali pertunjukan daripada obat modern asli Indonesia tersebut. Dan bahkan saya udah hitung di situ, di dalam buku itu, yang disebut sebagai Badan POM, yang disebut sebagai OMAI, OHT plus fitofarmaka ada 61 plus 24, kira-kira 85. Jadi cocok dengan apa yang tadi Dr. Terawan katakan. Ada lebih dari 80 obat OMAI loh ya yang sudah ada di Indonesia ini sesuai dengan apa yang sudah diterbitkan oleh Badan POM di Informatorium Obat Modern Asli Indonesia di mana ada 61 OHT dan 24 fitofarmaka. Jadi ini kita banyak sekali OHT dan fitofarmaka yang memenuhi persyaratan OMAI obat modern asli Indonesia. Nah, sekarang PR kita apa lagi? Bukan hanya membuat, jadi ini Pak Presiden waktu itu kami sempat bertemu, menunjukkan bahwa OMAI, Pak ini OMAI, ini harus kita lebih banyak lagi penelitian OMAI. Di universitas-universitas, termasuk di UNPAD, di mana saja universitas-universitas harus melakukan banyak sekali penelitian OMAI yang memang sudah didesain berdasarkan pharmaceutical modern dan juga pharmacology modern. Karena ini akan memenuhi persyaratan kita mengurangi bahan baku. Coba bayangkan kalau kita menggunakan salah satu obat diabetes saja dari herbal, berapa obat bahan baku yang sudah bisa diturunkan, importasi dan lain-lain ya. Jadi ini obat herbal terus fitofarmaka. Nah, by the way, yang ini kita lakukan ini banyak sekali sekarang sudah sebetulnya dari 24 fitofarmaka ini, kami sudah mempunyai 20 dari 24 fitofarmaka ini. Di sini ini sudah ada semua gambarnya ya. Saya tidak akan menunjukkan satu-satu, tapi ada cukup banyak obat untuk diabetes, ini tadi desmeno, yang tadi fraksi daripada DLBS 1442 yang untuk endometriosis, yang ini fraksi daripada kayu manis untuk mengurangi asam lambung. Ini bisa lebih baik dari omeprazol, kami sudah melakukan uji klinik, lebih baik dari omeprazol bahwa dia menaikkan pH dan lebih dari 24 jam dan lain-lain ya. Jadi yang ini baru yang kami luncurkan adalah ASI Morinol meningkatkan kualitas dan juga kuantitas daripada ASI ibu ya. Dan satu lagi yang saya tunjukkan bahwa semua produk Dexa termasuk obat kimia dan produk obat herbal sudah tersertifikasi halal oleh Majelis Ulama Indonesia. Jadi ini adalah salah satu peluang yang kita harus lakukan bahwa halal ini merupakan sekarang keharusan untuk produk di Indonesia. Sehingga pada waktu kami mendesain produk sekarang sudah dimasukkan jaminan halal di dalam suatu baku mutu di dalam pembuatan dan juga produksi daripada obat herbal tersebut. Obat OMAI, obat modern asli Indonesia tersebut. Jadi bukan hanya penelitian di lab dengan menggunakan enzim dan lain-lain, tapi bahan bakunya sudah harus dipastikan halal pada waktu kita mendesain daripada obat tersebut. Karena ini sekarang menjadi suatu keharusan. Ya. Nah, PR kita bersama adalah bagaimana OMA ini adalah formularium nasional, masuk dalam formularium nasional. Yang seperti Bapak Ibu sekalian, adik-adik sekalian ketahui, tidak ada satupun obat herbal di dalam formularium nasional. Kalau ditanyakan berapa obat herbal dari 7.000 tanaman obat Indonesia, tidak ada, nol, zero, tidak ada. Ini adalah tugas kita bersama, bagaimana mendorong obat herbal modern. Anda sekalian banyak sekali melakukan penelitian biodiversitas, tapi tidak ada satupun yang digunakan di dalam JKN. Jadi kalau Anda ke puskesmas minta obat herbal, tidak akan dikasih, karena itu tidak masuk dalam formularium nasional. Sehingga dalam hal ini adalah Bapak Ibu Maya Agustina Andarini ini adalah seorang deputi Badan POM deputi kedua pada waktu 18 Agustus lalu meminta bagaimana bisa mendorong dalam formularium nasional, ya. Belum ada satupun yang ada fitofarmaka obat herbal masuk dalam formularium nasional. Sangat disayangkan dengan negara yang begitu kaya, negara belum menghormati obat herbal yang kita peneliti Indonesia dapatkan masuk ke dalam formularium nasional. Sangat disayangkan sekali. Nah, oke, saya akan, ini kebetulan saya diwawancara di majalah Tempo tanggal 23 Oktober kemarin. Saya ditanya berapa lama sih menggunakan mencari obat farmaka yang tadi saya katakan. Rata-rata butuh 4-6 tahun, ya. Jadi memang banyak waktunya yang dikembangkan mulai dari praklinis kemudian sampai ke klinis. Itu memang lama, dan saya juga ucapkan keengganan dokter untuk meresepkan ini karena apa? Karena belum masuk formularium nasional. Sehingga edukasi harus dilakukan, termasuk di dalam pendidikan dokter dan lain-lain. Nah, Bapak Ibu sekalian, sampai terhadap ini slide yang terakhir, ini tempat kami melakukan riset obat modern asli Indonesia di Cikarang, dan ini adalah teman-teman saya semua yang melakukan penelitian obat modern asli Indonesia, dan akhirnya mari kita dukung OMAI, bangga buatan Indonesia. Terima kasih, salam sejahtera buat kita semua.
Okay, thanks, Mr. Raymond. Very interesting presentation, I think. We will keep a discussion session after the last one presenter present. The last one presenter is Mr. Sriwidodo from Faculty of Pharmacy Universitas Padjadjaran. Mr. Sriwidodo, are you ready to present? Okay, before we going to presentation, I will be brief CV of Dr. Sriwidodo. Dr. Sriwidodo is a one lecturer in the Department of Pharmaceutical and Pharmaceutical Technology, Faculty of Pharmacy Universitas Padjadjaran. He finished instructor program from Universitas Padjadjaran, locally, but I think best quality. His research interest in protein therapeutic, HTF, particle drug delivery, and I think he has published many articles within pharmaceutical, chemistry, and biological science. Okay, Mr. Sriwidodo, time is yours. Presentation time about 30 minutes.
Okay, thank you very much, Pak Ian, our moderator in this session. Do you hear my voice?
Yes, very good.
Okay, thank you. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. Before starting the presentation, I would like to greet our distinguished speakers and all audience attending the seminar in this Zoom meeting. We would like to share about research and development of extracellular secretion of human epidermal growth factor and formulation HEGF nanoparticle for chronic wound healing. It is a research collaboration between the Department of Pharmaceutics and Pharmaceutical Technology, Faculty of Pharmacy, and Department of Chemistry, Faculty of Mathematics and Basic Sciences. And it continues from my doctoral research. We divide the presentation into 5 parts. The first is the introduction, then the R&D strategy, the result, ongoing research, and the last is the conclusion and perspective. Next, please. Okay. In this slide, I will inform you the big picture of our project. We developed HEGF therapeutic protein from raw materials using extracellular recombinant. And then HEGF formulated into film-forming nanoparticle dosage form of chronic wound healing for diabetic ulcer application. Why we interested in HEGF? Because it has a unique structure and it is a potent mitogenic factor for the maturation of epidermal cells. To produce this protein, we can use recombinant DNA technology, the Escherichia coli. A host can produce HEGF by several advantages, including faster expression processes. We know there are two ways of protein expression using E. coli, intracellular and extracellular technique. When designing a recombinant DNA expression of HEGF, we consider three fundamental requirements for therapeutic protein: safety, quality, and effective. To fulfill the requirement, we have to get HEGF folded correctly and have high purity and biological activity. And the development of HEGF continued by formulating into a suitable dosage form, then testing the preparation to treat chronic diabetic ulcer wound to animal testing. And the scientific community is focused now on finding new treatment for wound healing and increasing therapeutic effectiveness. We are developing a new drug delivery system to release active compound HEGF in a controlled manner so that the management for ulcer is cheaper and more straightforward. Next, please. Okay, I want to describe the human epidermal growth factor in more detail on this slide. It is a single chain polypeptide composed of 53 amino acids. It has 6 cysteine residues forming 3 disulfide bonds, which support both the HEGF protein precisely. The HEGF protein weighs about 6.2 kilodalton. It has unique characteristic thermal resistance and not denaturated by heat to 70 degrees Celsius. HEGF also widely used as a healing agent for various chronic wounds and to regenerate skin cells and skin elasticity. This is based on 2070 data. The world diabetic mellitus cases recorded at over 425 million, and the prevalence of diabetic ulcer is around 15%, amputation 30%, and mortality reached 32%. Thus, we sincerely hope that the research and development of HEGF as a spray film forming preparation can help accelerate wound healing diabetic ulcers. Next. Okay, we have studied several recombinant protein investigation, especially HEGF, and finally found the advantages and disadvantages its expression techniques. On this slide, we want to explain that HEGF is more appropriate using extracellular techniques. We commonly use E. coli host cell because it has a quick growth process to minimize production cost. However, besides these advantages, the use of E. coli as a host cell has several disadvantages, including the accumulation of protein secretion becoming non-functional, high chance for the formation of inclusion bodies because it incorrect folding or misfolding, and the high rate of protein degradation because of their intracellular protease enzymes. The most effective solution to produce HEGF in extracellular protein through a membrane secreting into a periplasm or to the culture medium to overcome these drawbacks. And we introducing a E. coli membrane signal peptide on dependent peptide lyase B or PLB pathway become an existing strategy to produce secreted HEGF with proper free folding to generate high yields of active HEGF. The advantage using a secretion system in extracellular protein fold properly, and it makes biologically active, and it is a key point for our research and development. Polypeptides have authentic and terminals. Isolation and purification more simple. It not need for lysis cell, and higher stability and solubility because it already folded, free from endotoxin contamination because it excreted on culture medium, and the target protein is outside the cell and it's easy to purify. Next. Okay, we will move on to the strategy and our steps of the research we carried out. They are the extracellular secretion of HEGF using E. coli BL21, from optimization of the HEGF gene codon to verification and quantification of HEGF using ELISA. Next, upscaling to fermenter producing HEGF and purification. From production optimization using RSM to verification and quantification of IGF using ELISA. Then analyzing the activity and effectiveness of IgGF from testing cell proliferation activity using WST8 to in vivo wound healing testing. And for the last, formulation and evaluation of chronic wound healing preparation from formation of HEGF cytosine nanoparticle to evaluation and preparation. Next. Okay, I will present some result we have selected. This slide shows optimize the HEGF in codon that we did to get the optimal expression. Then construct the plasmid UDX61 with 4 different peptide signal, and codon adaptive index reach 1 value and percent GC 53.5, and that's good. Next. It is a cell growth curve. The 4 plasmid determined by the exact induction time at exponential growth. Then the post-induction harvest time determined at each plasmid highest cell density. Induction time range about 3 to 6 hours, and peak growth 5 and 8 hours. Next. Okay, this is the same slide. Next. I saw the result of the expression of the 4 plasmids in this slide. We can see that the characterization using SDS page shows that the 4 plasmid with different peptide signal express the HEGF target protein. It took samples from the culture medium at 8 and 18 hours. The approximately 6.2 kilodalton band, and calculating using a linear regression as shown in the figure. Next. In this slide, extracellular secretion have worked well. The evidence by the appearance of EGF band in the soluble fraction in cytosol and at the culture medium appear thicker, especially at 18 hours. Next. Here I want to show you something interesting. The 62 kilodalton or EGF band is translocated from the cytoplasm to the periplasm and exit the cell growth medium. It's so that the PLB peptide signal works well and carrying out the fully folded HEGF. Next. Okay, we confirmed that the HEGF target protein in the band of approximately 6.2 kilodalton was properly folded using ELISA. And you can see that the plasmid produced the highest HEGF, which is 310 microgram per milliliter. And moreover, we need to say that in another study, HEGF was verified using Western blot, and the 62 kilodalton band that appeared on SD's page also saw specific reaction on Western blot compared to standard and marker. And it is the beautiful picture that we have. Okay, next. This is the summary of extracellular secretion for your information. Next. We want to present the optimization step using RSM responsiveness method with a central composite experimental design. We take two main variables, the amount of medium and incubation time of induction with leveling -1, 0, and 1. There are certain run orders with a combination of software processing parameters, as you can see in this slide. Next. We carried out a 30 EGF extracellular secretion experiment on 200 milliliter flat scale, and we measured the amount of HEGF secreted using SPLC. We carried method validation out before sample measurement, and the highest retention factor value is 3641. Next. Optimization using RSIM based on the previous HEGF measurement result, then got a mathematical equation to see the relationship between the result and the optimal variables. We get the theoretical result close to the real HEGF result, confirming that the equation is valid. Then the base variable got from the RSM curve plot, and based on the amount of growth medium and optimal induction time, we obtain the highest HEGF result concentration, as you can see in the slide. Next. After obtaining the optimal 200 milliliter flat scale, we carried out a projection using fermenter scale. Using a batch feed system, you can see that the HEGF results are significant before and after optimization. And the yields of each EGF fermenter scale was 122 microgram per milliliter. Next. We perform HEGF purification utilizing the thermostable properties of the HEGF protein, and followed by ion exchange chromatography, and then the results are quite encouraging, reaching almost 82% in purity. Next. And it is a summary of production and purification of HEGF for your information. Next. We investigated HEGF activity, both in vitro and in vivo testing by looking at the cell proliferation activity using the WST method and Nih3T3 cell culture. You can see that HEGF increased the most extensive cell proliferation, 190%, and 50 nanogram per milliliter concentration of HEGF. Next. And we can see cell migration activity on this slide. It can be seen that HEGF at 50 nanogram per milliliter dose has accelerated the direction of strict closure on the Petri dish. Next. And we can show the result of HEGF effectiveness experiment on animal tests compared to negative controls. The test animals with HEGF administration of 50 nanogram per milliliter effectively accelerated wound healing of the mice. Next. And the epithelial cell parameter shows there are addition of significant fibroblasts relative to the control, and the indicator that HEGF is work well. Next. Okay, this is a summary of in vitro and in vivo testing for your information. I added information that acute toxicity test did not show any cell toxicity, and the endotoxin test with the LAL method, endotoxin doesn't appear. Next. Okay, we come to the last stage, the result of the HEGF formulation into nanoparticle dosage form. Here is the formulation we made and got significant result. Next. And the evaluation result based on parameters of the polydispersity index, pH, zeta potential, and absorption efficiency showed that the result met the pharmaceutical requirement, and the SAM and TEM morphology also showed a picture of HEGF encapsulation by chitosan nanoparticles. Next. This is a summary of formulation and evaluation. The optimization of nanoparticles, present transmittant is 98%, and the size is about 600 nanometer. The nanochitosan HEGF formulation with ion exhalation method, the IGF 79 nanogram per milliliter, and 1% chitosan, and sodium 3-polyphosphate 0.15%, and polyethylene glycol 2%. The characterization nanochitosan HEGF preparation size 600 and 800 nanometer, zeta potential is positive 33 and positive 41, and the same morphology shows the aggregate surface, and the morphology of them show the chitosan polymer matrix encapsulated protein. The evaluation of the preparation, pH in the range of 6 and 7, and organolepticality liquid, and entrapment efficiency reached over 99%. Next. Before to the final conclusion, let us convey the ongoing development regarding nanoliposome HEGF preparation. It's formulated by a water-soluble chitosan film forming system. Based on result of the evaluation that has been done, the preparation of the spray produce a spray angle and the occlusion factor meet for the requirement about 85 degrees and less than 15%, and the morphology of nanoparticle using TEM shows a picture of HEGF uptake in liposome. You can see in this slide, and we also make one dressing from chitosan water soluble. Okay, next. Next. Okay, this is our conclusion. Based on the result of the tests that have been carried out, HEGF has met the requirement for safety, quality, and efficacy, although it still requires a series of tests to complete the requirement. Nanoparticle technology based on chitosan and liposome is very suitable to water-soluble chitosan film forming preparation and feasible for chronic wound healing application. This collaboration research and development of HEGF therapeutic protein over the past 5 years has resulted in HEGF product, several scientific journal publication, patents for extracellular secretion method, and other IPRs. Next. Perspective, we are still developing chitosan nanoparticle and HEGF liposome into water-soluble chitosan-based film forming dosage form. We hope it increase stability and efficacy in their application. Extracellular secretion techniques have a high potential to be adopted in the pharmaceutical industry. However, it still needs development to achieve in economic value. And we've been doing this research for 5 years, and it's still ongoing. We realize that there is still a lot of research so that the pharmaceutical industry can take advantage of it. Next. Okay, this is the acknowledgement. And next. Okay, thank you very much for your attention.
Okay, thank you, Mr. Sriwidodo, for your presentation. Now we would like to invite to all speakers to join in discussion session. I think a lot question in chat room. Maybe you can read observed in chat room question. The first question from Dr. Yoga to Prof. Mary. Question is the fundamental issue of chemotherapy from cancer is sensitivity and selectivity to cancer cell and normal cell. How is it, based on your research experience in measuring a sample of natural active compound that have good sensitivity and selectivity? You can directly answer, Prof. Mary.
I have shown two mechanisms for chemopreventive properties or chemopreventive agents, and that is suppression of cancer as well as the blocking. Now, we can expect that a compound which has the capability to block would also produce a suppressing effect. Specifically, if a compound, that's why if you will look at the flow of the research that we had, we initially tested it first for antioxidant because that would already give the researcher a signal that it will have a potential chemopreventive properties like producing apoptosis and even preventing the proliferation of the cells. So my answer to the question is yes, it can both have the same mechanism in one particular natural product or extract. Thank you for the question.
Okay, how, Mr. Yoga, clear? Okay, next question for you, Prof. Mary, from Mr. Deden. I have two questions for you. What is the correlation between antioxidant and anti-cancer activity from a natural product? First one, and the second one, according to your research experience, which one will better directly measuring cytotoxicity activity and antioxidant in order to explore anti-cancer activity of natural product compound for beginning? Thank you. You can read in chat room maybe.
I'm checking on the question here.
Okay. The first question, what is the correlation between antioxidant and anti-cancer activity from natural product compound? Maybe you can answer one by one.
So the correlation of antioxidant and cancer chemoprevention or cancer itself will be the ROS, the reactive oxygen species. Because without the antioxidant acting on this reactive oxygen species, it would cause some damages with the DNA, proteins, and lipids. Similar with the diagram I have shown on the slides I have shown previously, that if there would be damage of the DNA, lipids, and proteins, it will eventually produce some effects such as hypertension, inflammation, aging, and leading to cancer. And it is always that you would see cancer would always start with inflammation. And we can assume that a compound with an anti-inflammatory effect is also a potential anti-cancer agent. And what is linked with that would be ability to scavenge nitric oxide, because nitric oxide is the one which produces inflammatory responses to our body once there is an increased production of nitric oxide, which is part of the reactive oxygen species that I have discussed previously. That is my answer to the first question. Can you repeat, please, the second question?
Okay. According to your research experience, which one will be better, directly measuring cytotoxic activity or antioxidant in order to explore anti-cancer activity of natural product compound for beginning?
Okay. Initially, since a compound with antioxidant will be a potential anti-cancer agent, what we tend to do is to look at its content first. I am referring to the secondary metabolites. That's why we initially start with presence of flavonoids and presence of phenolic components, because presence of these flavonoids and phenolic components would lead to a compound with an antioxidant effect. And with that antioxidant effect, we can now link it, an antioxidant compound will have the capability to scavenge these free radicals, I have given 4 free radicals as an example previously, the singlet oxide, hydrogen peroxide, nitric oxide, and also superoxide. In all of this, what is important is the hydroxyl ion and of course the nitric oxide, because they are related to inflammatory responses, which may lead to cancer. Thank you for the question.
Okay, thanks for the answer. Next, for question for Mr. Raymond. Question to Mr. Raymond regarding the concept strategy after choosing the gen or protein for specific disease. How do you choose which plant will be used for activity screening? And second question, what quality control must be done for the active action? Thank you. Maybe you can answer, Mr. Raymond. Mr. Raymond, you can join us.
Is already left.
Oh, maybe I will send your question via email maybe to Mr. Bayu Indradi. Maybe next question for Prof. Merizo again. Question to Prof. Merizo. Chemo prevention mechanism can happen in two ways. This on your slide. Block and suppress. Are a compound can have two way or prevent cancer, or only one way, either blocking or suppress? Based on your experience, natural product from marine have blocking or suppress cancer? You can answer?
Both. It's almost similar with the first question, that it can both block or suppress. The one particular active component can produce two mechanisms at the same time.
Yes.
Specifically, if it can block, we will expect that it also has the capability to suppress tumor or cancer. I also saw one question here about turmeric, sir.
Maybe I will one by one I will read. To Mr. Sriwidodo. Next question to Mr. Sriwidodo. What does the advantages of HEGF if used it to application into diabetic wound healing patient? Maybe you can read in chat room, Mr. Sriwidodo. What does the okay? You can directly answer.
Thank you.
From Dr. Ida Muspiro.
Okay, thank you for the question. In the diabetic ulcer condition, we can imagine the blood vessel do not reach the injured tissue cell because it blocks or damages, and this causes the cells to lack oxygen and not get wound healing factors in the blood. For the solution, we can giving exogenous HEGF, allow cell in the chronic wound tissue to regenerate cells. And based on clinical research conducted diabetes healing and wound care, using HEGF can improve the wound healing process in diabetic ulcer patient. That's my answer, Pak Yan.
Okay, thanks for your answer. Last question for you, Mr. Sriwidodo, from Dr. Yoga. Is the use of HEGF already on international market? Second question. What about the price if it's marketed, considering that our society is price sensitive? The third question, many questions, Mr. Yoga. How to ensure that during the production the HEGF there is no damage? Maybe you can answer one by one.
Okay, thank you for the questions. For the first question, I found HEGF topical preparation that had been produced by manufacturers. One of production is from Korea. I don't remember the brand name, but the existing product, however, the advantage of our product, we have developed and formulated into nanoparticle, which can increase the effectiveness and stability and the application. For the
second one, how about
question,
price?
The price offered by the manufacturer, I think we can make it can be competitive because the dose of IgGF is very small. It's around 20 and 200 nanogram, and we can produce HEGF in flash scale around 300 microgram per milliliter. So I think we can make competitive with other product. And last question.
Last question. How to ensure that during production the HEGF is not damaged? I think meaning how.
Okay, thank
for
you.
Safety.
It is fortunate that HEGF has a high stability to heat and an organic solvent because it has 3 strong disulfide bonds. And however, it is susceptible can damage with proteases. But with 3 disulfide bond, we sure the IgGF is very stable in the preparation. That's all. Ian.
Thank you. I think we can continue our discussion session. Discussion session is very effective, I think, because we have a limited amount of time. I think this meaning we have come to end of this discussion session. Okay, thank you for all participants and all speakers for your support for this session. I say thank you. Back to you, MC.
Thank you, Dr. Ian Sopian. We would like to invite Dr. Ian Sopian to kindly hand over the certificate to Professor Mary Joanna T. Corpus, PhD, Mr. Raymond F. Chandra Winata, PhD, and Dr. Sri Widodo. With all due respect, we now give the time to Dr. Ian Sopian.
Okay. On behalf of Komit and Dean of Faculty Pharmacy Universitas Padjadjaran, we will give you a certificate. We would like to thank and highly appreciate to all speakers for your participation and support of this event. Hopefully, we can collaborate and on next ISPST seminar. Certificate will be sent to you by email after seminar has been done. Thank you very much. Back to you.
Thank you, Ian. Thank you, Prof. Merizo.
Okay. Thank you, Prof. Merizo.
Thank you.
Distinguished guests, ladies and gentlemen, the next agenda is photo session. We would like to ask all of the participants to turn on the camera and take a picture together. We will count to 3 and then take a screen capture of the Zoom meeting. Due to the size of the screen, please hold your pose.
This picture, I think.
Okay, 1, 2, 3. Okay, thank you very much. The next agenda will be poster session. The poster session will be held at this online room. Thank you. Dear all participants, the next agenda is parallel session. You are kindly invited to join the Google Meet in the link on the screen and the chat room for the parallel session. After parallel session, the event will continue to closing ceremony at 12:30 p.m. The closing ceremony will be held at this Zoom meeting. Thank you. Distinguished guests, ladies and gentlemen, we have come to the final event of the 4th International Seminar on Pharmaceutical Science and Technology 2020. And it is my great pleasure and honor to welcome all of you again this afternoon. Distinguished guests, ladies and gentlemen, the next agenda is best poster and oral presenter announcement that will be informed by the chairwoman of ISPST 2020. With all due respect, we now give the time to Dr. Nyimakar Saptarini.
Thank you for your attendance during this seminar. I would like to announce the best poster presenter is Fit Haira Nusra from Universitas Muhammadiyah from Dr. Hamka. And the best oral poster is Wahyu Widowati from Maranatha Christian University. Congratulations to the winners. Hopefully it will be an encourage for the other participants. Thank you.
Thank you very much for Dr. Nyimakar Saptarini and congratulations for the winner. We are very honored to have with us here this afternoon Professor Mukhtaridi, PhD, as Vice Dean of Faculty of Pharmacy Universitas closing speech. Now, I would like to call upon Professor Mukhtaridi, PhD, to present us with the closing speech and ceremony.
Thank you to moderators, honorable of all speakers, keynote and invited speakers, excellencies, ladies and gentlemen of all our participants and committees also. Assalamualaikum warahmatullahi wabarakatuh. On behalf of Dean of Faculty of Pharmacy, Universitas Padjadjaran, and the fourth ISPST organizer, I would like to express my gratitude to all of you who have come together virtually from various regions, even various countries, for your cooperation, which has enabled us to conduct highly fruitful seminars. Now, we have reached the end of the fourth ISPST, and it's my task to provide some closing remarks. We actually organize the ISPST every 2 years since 2014. But due to the pandemic situation, we are forced to organize the fourth ISPST virtually. However, we hope that this seminar still has been conducted since yesterday is still beneficial for all of us. Once again, on behalf of the committee, thank you for delivering such important materials. Thus, all of us can deepen our knowledge about future perspective in the transformation of pharmaceutical research into applicable industry. We have gained a better understanding about the challenge to transformation of pharmaceutical research into applicable industry from the speakers, from university, academician, and from industrial. It's our hope that based on the face-to-face relationship established through the seminar, in the future, we will be able to respond together to issues that we have in common and achieve our common objectives. We also hope for your participation in the fifth ISPST in 2022. Excellencies, ladies and gentlemen, since the International Seminar Pharmaceutical Science and Technology in Universitas Padjadjaran is a biennial event, we look forward to seeing you again in 2022 when we shall reconvene to discuss another topical theme affecting the pharmaceutical science and technology. Finally, we would like to thank to the fourth of ISPST committees for their extraordinary effort to carry out the seminar. Again, thank you. Thank you all very much for your cooperation. Billahi taufiq wal hidayah. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you, Professor Mukhtaridi, PhD, for the speech. Distinguished guests, ladies and gentlemen, we have now come to the end of 4th ISPST 2020. On behalf of all the committee members and the university, I would like to express my utmost gratitude to all the distinguished guests and members for making this event a success. Now, I would leave you with a quote by Charles F. Kettering. There is a great difference between knowing and understanding. You can know a lot about something and not really understand it. Let us all be guided by all the things we have learned and heard throughout the seminar and be able to see and influence our future. Thank you very much, and please forgive us for any shortcoming. Till we meet again on the 5th ISPST 2022, and may all of you stay safe and stay healthy. Thank you.
Thank you so much. See you in the next ISPST.
Thank you for all participants. Meet you again in the 5th ISPST.
Inshallah in 2022, right?
Yes.